All right, before the break, I'm sorry, it's loud. Before the break, okay. We talked about antibodies can fight infection for us, and if you look on the right side, it's the antibodies that bind the free antigens. Free antigens may directly activate a B cell. That the B cells, they have these membrane-bound antibodies, and they can be stimulated to secrete antibodies when they become plasma cells. So for T cells, they don't work that way. They need a piece of antigen presented to it by another cell called APC. All right, T cells require an antigen presenting cell, or APC for short, to quote unquote present the AG antigen to it. Cells that have an antigen presenting function, there's not too many of them. Let me just list them now. They include macrophages. They include cells called dendritic cells. B lymphs, B cells can have this function. So those are the three that can be, that have the APC function. Now, there are different subpopulations of the T cell that I defined before the break. We're going to focus on CD4, CD8 cells. This is illustrating to you a concept called MHC restriction. It's the T cells that interact with the MHC, which I discussed last time. But there are different classes of MHC. There's class 1 as well as class 2. And different subpopulations of T cells are restricted to interacting with only class 1 or class 2. MHC, major histocompatibility complex, sometimes called human leukocyte antigen. Same thing. Well, anyways, I call it MHC. MHC restriction. All right, so they have the CD4 cells over there. Let's remember, those are the T helper cells. And they have the CD8 cells. Those are the cytotoxic killer T cells. And what we see is that CD4 cells, they mature, well, CD4 and CD8, both mature in, top of the figure, bone marrow. From there, they go to the thymus. That's where you have the 2 rounds of selection, right, the immunocompetent ones. And the CD4 cells, after they leave the thymus, they're restricted to interact with cells that present MHC class 2. CD8 cells are restricted to interact with cells that present MHC class 1. And then when they do so, the activation process will get our helper T cells, sometimes called regulatory cells. I tend to stick with helper T or CD4, and your cytotoxic T cells. So, and of course, like I said, you can get some cells that persist as memory cells. So here's an outline of that MHC restriction. I don't think there's any new information here that's not on the board here. So CD4, CD8 cells, they bind to different classes of MHC proteins. And, well, that's the restriction. The class 1 MHC, CD8 cells become cytotoxic T cells. They're activated by antigen fragments linked to the class of MHC class 1. In the class 2, the CD4 cells become helper T cells. They bind antigens linked to the class 2. So just have, it helps to commit that to memory. And basically, let's do class 1 first. And in studying class 1, class 2, I think it's good to ask yourself these questions to kind of keep it all in your head. So let's kind of go through it. When studying class 1 MHC proteins, know that they're recognized by the cytotoxic CD8 T cells. So we're doing this one first. Ask yourself these 3 questions. Firstly, which cells can present class 1 MHC? All cells with a nucleus. That's most of them, except for maybe like red blood cells. Remember, red blood cells don't have a nucleus. I mean, they spit it out right before they enter circulation. But okay, so that's the first, the answer to the first question. Which cells have MHC1? All cells with a nuke present MHC class 1. So the exceptions are RBCs, don't have it. They don't have a nuke. Platelets, any cell that doesn't have a nucleus will not have MHC class 1. But that's basically everything. Okay. The second question is, okay, well, they have MHC class 1, they interact with the CD8 cells, but what do they present? They present an antigen that's always endogenous. AG, antigen. That means it's a fragment, it's a piece of molecule that is presented. It came from within a cell. From within. Endo means in. So endogenous, it came from within. Now this antigen, most of the time, not sick, it's going to be a self antigen most of the time. So that antigen can either be self or non-self, but most of the time it's a self AG. You're doing fine, you're not sick, but if you get sick, maybe you get a virus or something, the infected cells may take a piece of that non-self and present it. So it could be non-self when you're sick. Be a piece of the virus or pathogen. It indicates there's microorganisms hiding in that cell, right? Virus infected cell, as an example. Well, here's a picture of that. To answer those 3 questions in picture form. Here they show the MHC class 1. Any cell that has a nuke is going to present MHC class 1 to interact with your white blood cells, the CD8 cells, right there. But what you do is take a piece of self, let's say you're not sick, that fits in that groove, and then as your white blood cells circulate by, the CD8 cells, they recognize class 1, they don't react with self AG, and they just keep on going. Okay, everything's working fine. let's have the same conversation for class 2. Which cells present class 2? It's the APC cells, right? Dendritic macrophage B cells. Answer the first question, which cells have MHC? APC cells present MHC class 2. What do they display? They display exogenous antigens. So something that they've phagocytized. Because it's something they phagocytize, it's exogenous. Because remember, those are your white blood cells. Because it's always something that's exogenous, it's always going to be non-self. So this is that first step of, recognition. Can you recognize self from non-self? So first of all, this APC cell recognized this is non-self. The garbage, the pathogen, the bacteria, whatever it is. And it engulfed it, okay? And then, part of the recognition process is displaying a piece of that and putting it in the class 2 to the CD4 cell. Okay, so recognize here, this cell recognizes it, display it here, so that the T cells, the T helper cells, have a chance to recognize it as non-self and mount a reaction. So immune system reactions start with the T cell activation of the CD4s. Okay, and then the CD4s kind of activate everything else. So let's start by talking about T cell activation of the CD4s in 3 steps. The T cell receptor has got to recognize the MHC class 2. The T cell receptor has got to be specific for that particular viral antigen, and CD4 stabilizes it. But there's also co-stimulation. That's important too. So here's a picture of this outline on the next slide. Let's kind of draw it out. So it's like you're sick, you mount an immune response, you start with the CD4s. They're the linchpin, they kick everything else into gear. I think help is a good way to describe them. They're helper cells. They help everything else get going. I said they're the linchpin. I don't even know what a linchpin is. I don't know why I said that. They're helper cells. They help everything else. So you got that APC cell. Could be a macrophage, dendritic cell. It presents the MHC class 2. I'll just put 2. That's MHC class 2. So APC on this side. What it's doing is it's interacting with the CD4 helper T cell. Oh, I forgot. Let's put the viral AG in this group here. You got the helper T cell, CD4 cell, whatever you want to call it. It presents a T cell receptor that can interact with MHC2, but it's also specific for this viral antigen. So TCR. Okay. Now there's a cell differentiation protein, CD4, that fits in that groove that helps stabilize this. So the cell is named for this surface protein, CD4. Just call them CD4 cells. It fits in that groove of MHC2. And this triggers a cell signal. And you get this specific binding, TCR to MHC2, specific for the viral AGs. Call it signal 1. To prevent unnecessary premature activation, you actually require a second signal. So, well, I'm just going to draw another ligand here. This is CD28. And it interacts with another ligand called B7. So this CD28 B7, that's a co-stimulation. It'll trigger a second signal, call it signal 2. But you need both signal 1 and signal 2 to really facilitate, well, let's just say you complete activation, but you need both signals, okay, with the co-stimulation. Complete T-cell activation. The other thing is interleukin 1 also helps this activation thing. So it goes from APC to T-cell, interleukin 1. And they named the molecule something very easy to understand. It's a molecule secreted between two leukocytes, interleukin. It helps the activation process. That completes the activation, and a cell that has been activated, it'll present differently. For example, an activated one, it'll upregulate inflammation and further activate other APCs. You start to see other cell differentiation markers like CD154. That's after it's activated. Okay, upregulate inflammation, activate other APCs. It'll secrete something like IL-2 to help other things get activated. So IL-2, CD154, that's after you're activated. Okay, so now that the T helper cell gets activated, let's kind of look at what it does to other cells to help them. To emphasize the importance of co-stimulation. If you don't have that second signal from co-stimulation, energy occurs. Where the T cells, this means, wah, 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 you don't do anything. So you need that co-activation. So before I proceed, let's review this right there. So we said in this outline, you need, for T cell activation, number 1, you've got to recognize MHC2. Right? So CD4 interacts with MHC2. Check that. Number 2, TCR is specific for this viral AG. This particular clone has to recognize that particular, well, I drew it as a red ball. That's step 2. And I said CD4 stabilizes this. Thirdly, co-stimulation, B7CD28. Okay? that'll get you signal 1, signal 2, and that completes the activation. And I also mentioned interleukin 1, so I got all the things in this outline illustrated, so let's move on here. The helper T cell, here's how it helps an APC and a CD8 cell. So there's the helper T, APC, CD8. So what's it's doing, let me clear this out. So the helper 2 is secreting IL-2, let's see here, to help stimulate the CD8 cell. So that's the third point. Dendritic cell, let's see. Oh yeah, okay. It's activated with the help of interleukin 2 by the helper T cell. How do the activated helper T cells help? That's the question I'm answering. How do they help? Well, IL-2 helps to activate CD8 cell. Those become the killer cells, the cytotoxic T cells. Also, what you see here is the T helper cell is helping the dendritic cell to express those co-stimulatory molecules, the B7 ligand. Helps the dendritic cell express co-stimulatory ligands. And I mentioned B7. There's others, but that's the one I used today. Okay, great. There's also help of the B cells. So here you see the T helper cell. It binds with the non-self complexes of a B cell that has encountered its antigen and is displaying MHC2 on its surface. So the T cell releases interleukin 4 as a co-stimulatory signal to complete B cell activation. So I guess as a third thing, 1, 2, 3, helps complete B cell activation via IL-4. I want you to at least see one slide of the CD8 cells in action, the cytotoxic T cell. When they're activated, what they can do is they target virus-infected cells and administer a lethal hit. That's what's shown here. Activated CD8 cells target virus-infected cells and administer a lethal hit. So on that slide, read through it. What you want to do is look at the perforins and granzymes. That's kind of how they're killing the cell. I'll just go to here. Perforins, granzymes. That's the quote-unquote lethal hit. Perforin molecules insert into the target cell membrane, polymerize, and They have these transmembrane pores, just cylindrical holes. Similar to those produced by complement. Granzymes then enter, right, the target cell via the pores, and what's inside? Proteases degrade cellular content, stimulating apoptosis. So that's the lethal hit. I had some practice questions here. We don't need to do those now. I know you're thinking about your blood typing test. So I'm going to stop here. And you have the rest of the time. You won't need it all. We're going to go right in to the test. I'm not going to reseat you, but I'll just ask for right now. I got to go grab the tests in the other room. Just clear your desk, get dividers, put it between yourselves, and when I come back, I'll just pass it out.